All right, Assalamu alaikum, everyone. I am Farah Abul Rub. I'm a third year medical student, and for today, we are going to be going over the second part of our immunology crash course, where we will be covering the last three portions of our contents, which are the complement, MHC, and antigen presentation, as well as some practice questions. Before I get started, do you guys have any questions at all about the previous three lectures that we have covered? Going once. All good. All right, perfect. Okay, so before I get started on the compliments, I would like to take you guys back to our play and just try to remember some of the characters that we had, which are going to be relevant during today's session as well. So our play unfolds, and we have those two boys right here. How old are they? Because can anyone help me remember? How old is this one right here? 4 and 8, exactly. Perfect. Okay. So what does the 4-year-old like to do? Help her T cell. Okay. Help his brother and help his. Who else does he like to help? Activate the B cell. B cell macrophage or just the B cell? Because a macrophage and a B cell are two different things. Babysitter. Yes. Perfect. Okay. So the B cell. And then what about this 8-year-old? What does he like to do? Cytotoxic T cell, exactly. So he always wants to protect his family, right? All right, our next character was her. And who was she? How old was she to begin with? B cell 19. Okay, perfect. So she was 19 to 20 years old. And what was her role? How was she related to those boys? She has a presentation due, yes. She always has a presentation due. But how is she related to those two boys? What is she to them? And what gives her her name? Because she's a B cell, so she is a. Guys, come on, wake up. She's the babysitter. Okay, our next character. How old was this guy? He was 56, exactly. Okay. And with all of those extensions coming out of his hair because he's continuously anxious, he is a dendritic cell. Exactly. So those three cells are our main characters for today. However, we are going to be having other characters as well that will be taking part of our session. So let's take a look at those. First of all, we have this very hungry boy. How old was he? 14, exactly. Okay, so he's always hungry. He goes sleepwalking unless he's woken up as a monocyte, and then when he is woken up as a macrophage, he has multiple nicknames because all over the body he is called different things, which is within your first lecture. Like, for example, within the brain, it's a microglial cell, within the liver, it's a Kupffer cell, so on and so forth. Next, we have this character. The reason why she's yellow is also because she is the main component of pus, which I'm not sure if you guys have covered yet in pathology, but it's a nice thing to link. How old was she? 15 years old. And why does she have those freckles? Granules, exactly, because she is a granulocyte, along with the many other characters that we have described last time, which are part of her family, right? She's a germaphobe because it's the first responder for a bacterial infection, and she likes fishing because neutrophils often create nets. Lastly, we have this very buff dude who was our bodyguard. He is 100% natural and he can easily detect a catfish. This is our, what type of cell? Natural killer cell, exactly. So he's 100% natural as a natural killer cell. Okay, without further ado, let's us get started on our first lecture of the day, which is the complement system or the complement cascade or the complement pathway or whatever you'd like to call it. So the complement system is basically a collection of more than 30 enzymes and proteins produced by the liver and it's released into the serum, much like almost every protein that we have in the body. Okay, so they play a key role in clearing the bloodstream of immune complexes. Not all of these enzymes are involved in the activation of complement pathways. Some of them are responsible for preventing inappropriate complement activation. These types of enzymes are known as acute phase reactants, and those are very important in identifying certain inflammatory pathological diseases. So our complements are going to interact with both the innate and the adaptive immune system. However, most of them are secreted as zymogens. Do you guys remember what those are? Zymogens are basically what I told you guys to relate to and the repro with are the pre-pro proteins that we have, right? So they are inactive until they get cleaved, and then afterwards they are able to become functional enzymes. And the example that we had for that in our last session was interleukin 1 and interleukin 18, which are zymogens until they get cleaved by the inflammasome. That is a collection of the node-like receptors. All right? So our complement proteins are a series of proteins marked with C for complement that range from the numbers 1 to 9 based on their order of discovery, not their order of activation, which can get somewhat confusing, but thankfully, once you get to a certain point, they do follow the actual number scheme as well. So in order for us to understand the naming system of them even better, I want you guys to think of it this way. Whenever I put an A after the number, so let me say C, then 3, so I have the third complement protein. If I put an A after it, that means this is an anaphylatoxin. Do you guys remember when you took in first year an anaphylactic shock? Do you remember what that means? Hypersensitivity, all right, perfect. So an anaphylactic shock is basically when you do have hypotension, that's true, but the main point of an anaphylactic shock is a very severe allergic reaction. And what happens when you have an allergic reaction? We have this vasodilatation, we have this recruitment of inflammatory cells, we have this release of cytokines, all of which, as I mentioned last time, are basically chemoattractants. So we have chemokines and cytokines that lead to this chemotaxis, where we are pulling in cells from the body to reach this area. So when I have this A subscript afterwards, like when I say C3A, I am referring to an anaphylatoxin. I am also referring to the smaller portion of the protein. However, if I put the B subscript after, I am talking about the binding protein or the larger subunit of it. However, you're in medicine, nothing's going to be easy. So the complement protein 2 follows the exact opposite. When I'm talking about the big portion, I will be using A, and when I'm talking about the small portion, I will be using B. All right? It's just for C2. Everything else follows the same convention. So C1, C3, 4, and onwards all follow the same one. All right? These complement proteins together, they are going to begin a complement cascade or a series of events. And I want you guys to think of that basically like a domino effect. If you were to tip the first one over, it is very unlikely for you to be able to stop this series of events. And you guys have answered me very beautifully about what an anaphylatoxin is, and it is an inducer of hypersensitivity. Okay. Now, who of you can recognize this character? This is the newest addition to our play, and he is the main character of the compliments. Macrophage, I understand why you'd say that, because he looks pretty hungry, but I'm talking about the actual characters. So Pac-Man, that is perfect. Good job. But instead of us using Pac-Man in this lecture, we are going to be actually using MacPan. And I personally like to do things like this. It's somewhat funny, just confusing people, walking around being like, oh, that's MacPan. However, every letter for this will actually stand for something that's going to help us understand this better. So Mac is the final target or the final goal that we want to achieve from the complement cascade. We have multiple different things that can happen, but this is usually what we're working towards. So Mac is going to stand for membrane attack. In order for us to reach this membrane attack complex, we're going to have three different pathways. It says my internet connection is unstable. Is my voice cutting up, or is everything okay, guys, on your end? I'm good. All right, can I just get a confirmation from one more person? All right, perfect. Okay. So this membrane attack complex is going to be reached through one of three pathways, either going to be triggered by a protein, an alternative pathway, or a normal pathway. Now, those aren't exactly what they're called, but it's just a way for you to remember the overall picture. Now, let's get started. As I mentioned at the beginning, this is my favorite lecture from immunology, so it took me a very long time to do this, but I hope it's as fun for you guys to go through as it was fun for me to make. I drew everything myself, so hopefully it's not that awful to get through, and hopefully it makes sense. So first of all, we will be going through the classical or the normal pathway based on our mnemonic. First of all, we are going to be having some kind of pathogen. In this case, it's a bacteria. And to this bacteria, this type of structure is going to attach. What is this? Based on the way that it looks, can you guys figure out what this structure is? An antibody, perfect. So an antibody, usually IgG or IgM, is going to bind to this pathogen. Once it does that, a type of protein, which is going to begin this complement cascade, is going to attach to the Fc region on this antibody. This protein is called C1q. So I know I didn't mention to you guys Q, so what is Q? Well, C1 is also unique in the sense that rather than it having an A and B, it has three subunits, Q, R, and S. So once C1q binds to the Fc region of this antibody, it is going to start to cause a serine phosphorylation of the remaining subunits, starting with C1r and then followed by C1s. Now, all in all, together, this is going to be called C1qrs, or for short, you can say C1s. So far, so good. Are you guys following along? All right. After we have this C1QRS or our CS, C1S, sorry, doesn't it kind of look like a hammer to you guys? So based on that, it must have some kind of function where it smashes things, right? So it is going to work on our next complement protein, C4 and C2, in order to cleave them. So are the C's complement factors? Yes, they are the complement proteins. So this C1QRS is going to work on our C4 and C2 to cleave them into their smaller and larger subunits. So as I mentioned to you guys, since this follows the normal convention, the larger part should be, actually, I'm sorry, the naming here is switched, but the larger part should be the C4b. Those are switched. It should be C4b, and the smaller one should be C4a. And then for this one, the larger part should be C2a, and the smaller part should be C2b. All right? Once it has cleaved those into the larger and the smaller portions, this C4b is going to bind to the C2a because those are the binding portions, right? I told you guys B is for binding, A is for anaphylatoxin. So this anaphylatoxin portion is going to go away. It's going to go to the bloodstream and be an anaphylatoxin. It's going to recruit other types of cells to the site. But this B portion is going to do the binding, at least for the B, at least for the 4 complement protein. For 2, it is the other way around, so we're going to be using the C2a. Given that, we will be forming this type of protein, which is our... Did I write this wrong? I think I wrote this wrong. Yeah, I'm sorry, I wrote this wrong. It should be C4b2a. I'll fix this in the presentation I actually send you guys. All right. However, what's really important is that this is our C3 convertase. Very convenient name, in my opinion, because if we take the C3 convertase, what do you guys think it's going to act on next? C3, exactly. So this protein right here is our C3. C3 is going to bind to the C3 convertase and be cleaved once again into C3b and C3a. C3a is once again an anaphylatoxin that will get released and it will recruit cells. However, the C3b is going to bind to the C3 convertase in order to give me the C4b2a3b, which is also known as a C5 convertase. So I had a C3 convertase and a C5 convertase. That is so much more important for you guys to remember this name than it is the actual entire length of the name. Because in the exam, he might ask you about those two. All right, we have a C5 convertase. What are we going to make it act on next? C5, exactly. Okay, so our C5 convertase is going to bind the C5 and it's going to cleave it into C5a and C5b. C5a, anaphylatoxin, C5b, binding protein. The C5b is going to start binding to C6, C7, C8, and C9. And don't those kind of look like something I showed you guys in the beginning? Every single one of them? They kind of look like our MACTAN, don't they? They kind of look like our membrane attack complex. So once we have this binding of C5b to C6, C7, C8, and C9, we finally have this formation of a membrane attack complex. So going over this again, would you guys prefer I correct those so you don't memorize them the wrong way? Like right now, just so you don't keep looking at them this way? All right, just a second. I'm sorry, I hadn't noticed the first time around. Just give me a second. I apologize for that, you guys. I had not noticed the first time around. Okay. Now looking at the full picture again. This, just a second, let me get my pointer. So looking at the full picture again, we're going to have a pathogen that's going to get bound to by an antibody that's either IgM or IgG. The Fc region of that antibody will get bound to by C1q, which will lead to the phosphorylation of C1r and C1s, giving us the C1QRS complex. The C1QRS complex is going to cleave C4 and C2 into their smaller and larger subunits. C4A is going to be the smaller one, C4B is going to be the larger one. C2A, however, does not follow this convention, so it is going to be the larger one, and C2B is going to be the smaller one. These smaller proteins are going to be secreted as anaphylatoxins, and the larger ones are going to bind together to give me this type of structure here, which is my C4B2A, also called my C3 convertase. C3 convertase is going to act on C3 to cleave it into C3b and C3a. As C3b binds to C3 convertase, it becomes C5 convertase. C5 convertase will cleave C5 into C5a and C5b, which will go on to activate the membrane attack complex by binding to C6, 7, 8, and 9. Is that clear for everyone? Do you guys have any questions at all about the classic pathway? Because once you understand this one, you have basically done all complements. All the rest follow the same exact thing. I made it so clear. I'm really glad. I really apologize, guys, for the confusion. I had not noticed, but I hope that now it is clear at least. All right, perfect. So we can move on to our next pathway, which is our protein pathway. Our protein in this case is going to be our mannan-binding lectin or a ficolin type of protein, where instead of the immunoglobulin activating the entire cascade, the mannan-binding lectin itself will be the structurally similar analog to C1 and the immunoglobulin. So it's going to take up both of their functions. So when we have this pathogen, this mannan-binding protein that looks so similar to C1, as you guys can see here, is going to bind to the pathogen. It does not need an immunoglobulin because it is the colors of the immunoglobulin, so it takes over its function, and it is the same shape as the C1, so it takes over its role as well. All right? So it is structurally similar to C1, and so it's going to do the exact same thing, where it's going to cleave C4 and C2 and give us C3 convertase. C3 convertase, once it's bound to C3, is going to give me what? When I bind C3 convertase to C3b, what do I produce?
C5 convertase.
Perfect. Okay, thank you so much. So someone wrote out the entire thing, which is completely correct as well. C4b, 2a, 3b, which is known as C5 convertase. C5 convertase will act on C5 to cleave it into C5a and C5b. C5b will then bind to C6, 7, 8, and 9 to give me my membrane attack complex. Now, I mentioned the membrane attack complex, but how does it actually work on the cell in order to kill it? Well, the membrane attack complex, as you guys can see, our MAC pan here, is going to sort of bite off this bacterial cell. And once it has that hole in it, a bunch of fluids in the body are just going to rush into it, right? And think of a balloon that you just keep on filling up with water and water and water and water. Eventually, it is going to burst. So that is exactly what happens. It's going to bite off this bacterial cell, and fluid is going to rush in. Eventually, it's going to swell up and just die. So the cell will lyse itself. Is that clear for everyone? All right. Lastly, we have our alternative pathway. Now, since I'm mentioning alternative, then something has to be different, right? So far, we have had something that has to activate C3 in order to cleave it to C3b. We have had to had a C3 convertase, right? Because C3 has been our main character through all of this. However, in an alternative pathway, you don't actually have anything that activates C3. C3 activates itself. Now, typically what you have is this type of infection where C3, complete C3, is going to bind to the pathogen. After it does that, it is going to cleave itself into C3b and C3a. Once again, C3a, anaphylatoxin, C3b, binding protein. The C3b is going to bind to protein B. It wants to bind to it. However, the configuration of this protein B doesn't really fit in with C3b, right? It doesn't match. So we need something to once again cleave this protein in order for it to be able to fit in with this C3b. The protein that does that is called protein D. As it cleaves protein B, once again, it's going to give us an A portion and a B portion. So this will be protein BB. I understand this is confusing, but try your best to follow along with me. When this protein BB is going to bind, I'm going to feel like I'm rapping very soon. We're going to have C3BBB, which is also our C3 convertase in this specific case. However, looking at our C3 convertase, this protein BB still does not really match up pretty well. There's still this space, right? So it's not completely stable. In order to stabilize it, we are going to add propyridine or factor P. So what does that give us? C3b, BB, P, right? Now, with our stabilized C3 convertase, we can move on and try to give us a C5 convertase. Now, what did I use to add to a C3 convertase to make it a C5 convertase? C3b, exactly. So this C3 convertase will continue to cleave C3 and produce its own C3b that it will bind to. Once it does that, you have a fully functional C5 convertase or a C3b, B, B, C3b. All right? Does that make sense for everyone? All right, perfect. So, going over everything again, very briefly, let's look at our classical pathway. You're going to have IgM or IgG that's going to simulate our C1QRS. Our C1QRS will cleave C4 and C2 to give me my C4b2A, also known as, what's the other name for this complex? C3 convertase. Perfect. So C3 convertase will cleave C3 into the smaller C3A and C3B. Once I add C3B to my C3 convertase, I'm going to have my, what type of enzyme is this going to be? What type of complex is this? C5 convertase. Perfect. My C5 convertase will cleave C5 to give me C5b, which will then bind to C6, 7, 8, and 9 to give me my MAC-PAN. The protein pathway that I told you guys about, or the mannose binding lectin pathway, is the exact same thing, except we don't need an antibody and we don't need our C1QRS because it is structurally similar to C1QRS and it binds in place of the antibody. In our alternative pathway, we don't even need anything to activate our C3. C3 will, in and of itself, bind to our pathogen, split itself into C3A and C3B. C3B will then start looking for something to complete its catalytic activity. It finds factor B, but factor B does not fit in with our C3b. So factor D will cleave it into factor BA and factor BB. Factor BB will bind to C3b, giving me C3b-BB. Factor P will stabilize this complex, and it will be known as I don't know if it's my internet or you guys haven't. Okay, there we go. C3 convertase. Perfect. When I bind a C3b to a C3 convertase, what am I going to get again? A C5 convertase. C5 convertase will work on C5, cleave it into C5b, which binds to C6, 7, 8, and 9 to give me my membrane attack complex. Is that clear for everyone? I want you guys to be so good with compliments that you don't even have to go back to the lecture. I want this to be perfect in your head. If you guys need me to repeat, I don't mind repeating the entire thing again, every single pathway. If you have any questions, please let me know. Even if it is clear, but you'd like me to go over it once again, just so you memorize it better, that is completely fine by me as well. All clear, okay. And the alternative pathway, isn't it C3b, BB, P, C3b? Yes. So I have never found anything that includes the P except Dr. Garwin. So just remember the P as being there. But this picture, it does not include the P, and I haven't found it in any other infographic either. But the P is basically just something to support it and to keep it stable. It's not the actual C3 convertase. The actual C3 convertase and the alternative pathway is our C3b, BB. All right, perfect. Any other questions at all? Okay, so let's move on then. And let's look at the bigger picture. I kept on telling you guys about this anaphylotoxin. I kept on telling you guys, oh, they're gonna go do something. But what do they actually do? Like, I'm just not gonna throw them away, right? The body's very resourceful. It uses everything that it actually makes up. So C3A, C4A, and C5A are acute inflammatory cytokines. They're going to cause inflammation by binding to the mast cells and causing them to degranulate. Do you guys remember our mast cell character? She was the one with a lot of freckles and curly hair. This degranulation of hers is going to release histamine, and this causes this vasodilatation. And that is what gives them their name, because they basically act as if you're having an allergic reaction or an anaphylactic shock. So they're going to secrete these, they're going to cause the secretion, sorry, of this histamine, they're going to cause this vasodilatation, and they're also going to be working as chemotoxins, which can attract neutrophils, lymphocytes, and other immune cells to that inflammatory site. Now, remember, when I'm forming an attack complex, a membrane attack complex, everything is sort of stuck together, right? So I'm not going to be moving away from this pathogen. I'm going straight to it. However, I might need some help. I might need the immune system to jump in and help me out. So those small portions of the protein, those anaphylatoxins, are going to go and be chemotaxing agents, where they're going to be recruiting neutrophils, lymphocytes, and other immune cells. Also, aside from just forming the membrane attack complex, we can also tag this pathogen as an opsonin. Now, if you guys remember from our last session, opsonization is basically the process upon which a macrophage is going to detect the Fc region of an antibody bound to a pathogen. So the pathogen gets met with an antibody, the antibody binds to it, and then the macrophage has an Fc receptor that is going to recognize that Fc region and realize, okay, something is wrong. An antibody should not be binding to this if it was a normal cell. The same exact thing happens with C3b and C4b. Natural killer cells, macrophages, neutrophils, B lymphocytes all have receptors for C3b and C4b. So they work as opsonization proteins or opsonins. All right, so think of the cops. So those are basically the ops. They just go and they tag something, and then the ops are going to come, they're going to get rid of it. As I mentioned to you guys, we're going to have a recruitment of inflammatory cells, opsonization of the pathogen, or killing of the pathogen immediately using our membrane attack complex. So since this is a pretty aggressive type of response, we need to have something that is able to regulate it, right? We can't just keep this on the loose, because what if the C3 decides to bind to a regular body cell? What if our mannose binding lectin just decides, oh, I don't like the cell, I'm gonna start a full-on complement activation cascade in order to get rid of it. And as I mentioned to you guys, a cascade is like a domino effect. It is very difficult to stop it once you have started it. So we need to have those regulators that put a pause when we don't need this type of reaction to happen. First, we're going to have a C1 inhibitor that stops the complement activation early. Now, a C1 inhibitor is going to mainly work on which of the pathways that I mentioned to you guys? The classical pathway, perfect. Next, we have our decay accelerating factor, which is a lipid-linked cell surface protein that disrupts the binding of protein BB to C3b. Where did I need binding of protein BB? And which type of pathway? The alternative pathway, perfect, okay. Now, factor 1 can proteolyze and inactivate C3b. So which pathway would this work on? All of them, yes, perfect. Because if we don't have C3b, we're not even going to be able to form a C5 convertase, right? We can bypass the C3 convertase steps that we have in the alternative and the protein in the classical pathway, but we cannot possibly bypass the C5 convertase step of adding C3b onto C3 convertase. So we need it for all the pathways. CRI or CR1, sorry, it's the final one, and it can displace C3b and promote its degradation. So CR1 is very prevalent on reticulocytes or erythrocytes, and it's present on the red blood cells. All right. So when I'm looking at a red blood cell, it's going to have this complement receptor 1 that is going to bind this C3b, displace it, promote its degradation. And then it's basically going to send the immune complex to the liver and the spleen, and they're going to get phagocytosed by macrophages. Does that make sense for everyone? All right, perfect. And that brings us to the end of the first lecture. Do you guys have any questions for me at all? Since this is a checkpoint, feel free to even unmute yourselves and ask me. I do not mind whatsoever. If you guys need us to take a break, I don't mind either. All good, okay. Would you guys like to take a break, or should we push through? Should we keep going? All right. So if I ask you guys any questions about complements now, you're gonna be able to answer it, right? You know everything about them. You're naturally both. Okay, so you're a natural killer cell. All right, so let's move on to our next lecture, which is MHC and antigen presentation. This is the last lecture that is included in your midterm, so hooray, good job everyone. You have finally made it to the end. Let's get started without any further ado. So, do you guys remember who those were? We just went over them. Four and eight, B and T cells. Perfect. So they are our lymphocytes. So first of all, I don't like the fact that this lecture is included in you guys' midterm because I feel like it's such an abrupt shift into the adaptive immune system and T cells when it really should have been just that complement. But either way, we can move from there. The other end of our adaptive immune system, after we have already discussed the antibodies involved with B lymphocytes, after we have discussed everything that we just did, we are going to start looking at the T lymphocytes. And they're called T lymphocytes because they emerge from the thymus. Our B lymphocytes are called B lymphocytes because they emerge from the bone marrow. So all our T lymphocytes come from a common CD3 positive progenitor. And then they get divided into CD4 positive and CD8 positive. Now, what is CD? And I just keep on mentioning CD, CD, CD. What is CD? It's not a record, obviously, but it is a cluster of differentiation. They're basically like the tag on every cell in the body. So when you're trying to go and search for a specific type of cell, this is especially important in cancers, for example. If I have a cancer of the lymphatic system, I want to try to find out which type of cell is causing me this presentation. And I'm going to start using something known as a flow cytometer. I saw in your transcription that Dr. Garwin said not to know the details of it, so I chose not to include it in the session. But a flow cytometer is basically a cell counter that is going to use a bunch of different rays of light that are going to count each cell individually, as well as look at the markers that this cell has and this cell is presenting. So these CD antigens are what makes us know the type of cell that we're looking at, and every single cell in the body has them. I told you guys that our B cell is CD19, CD20, CD10. Our T helper cells are CD4. Our cytotoxic T cells are CD8. Our dendritic cells are CD56. So every single cell in the body has its very specific type of marker. If they come from the same origin, they're going to have a shared marker, which in this case is our CD3 antigen, okay? Is my voice cutting up again because it's saying internet unstable, or is everything fine? All right, perfect. Now, our CD4 positive T helper cells are going to recognize antigens presented on MHC class 2, and our CD8 positive cytotoxic T cells are going to recognize antigens presented on MHC class 1. What is MHC? I mentioned this to you guys in our last session pertaining to a specific type of cell that reacts to absent MHC. What type of cell was that? Can you guys remember? What type of cell in our body recognizes an absent MHC? Exactly, our natural killer cell. So what was MHC? It was our major histocompatibility complex. And in humans, it has a different name, which is human leukocyte antigen or HLA complex. It is the exact same thing. Okay? Our major histocompatibility complex is basically going to have classes 1 and class 2, which I told you guys CD4 positive and CD8 positive are going to have different receptors or different MHC markers. And I mentioned CD4 with 2 and CD8 with 1. So think in your head that when I multiply them, the sum or the product, sorry, always has to be 8. All right? So 8 times 1 is 8, 4 times 2 is 8. And I always have to be sure that a T helper cell or a CD4 positive cell is only ever going to react to something presented on MHC class 2. And the same goes for a CD8 positive, which will only respond to something presented on MHC class 1. So MHC class 1 are basically the barcodes that you guys have on every nucleated cell in the body. So RBCs are not going to have MHC because they are non-nucleated cells. And this class of MHC is basically what determines whether you can accept a transplanted organ or not. And why is that? Because when I have the cell on every type of cell in my body, on every nucleated cell in my body, this is my barcode. If my barcode does not match up with someone else's barcode, they're going to immediately recognize this and be like, no, this isn't myself. This isn't what I should be seeing, right? Because this is what is normally there. This is what should always be there. This typically is going to be expressed when I have an intracellular pathogen, like a virus, for example. And cells won't usually express MHC unless they're loaded with an antigenic peptide, because there is no anti-expression of MHC molecules. However, they're basically going to have degraded proteins that are being recycled to make new ones, and they're just presenting those. So you always need to have some sort of MHC presentation, but the way that it's presented is very different. I either have an antigen or I have just a degraded protein that is showing like, okay, I'm fine, I am normal, nothing's wrong with me, because I can't have an absent MHC entirely, as I mentioned to you guys, due to our natural killer cell. Our class 2 MHC is going to recognize antigens presented on professional antigen presenting cells. And that includes this guy right here. Who was this? Our macrophage, exactly. What about those two? Who are they? CD14, perfect, yes. What about those two? She looks like she's giving you a side eye, but that's just the bullet point. Dendritic and B cell, perfect, okay. Now, with regards to the different subtypes that we have, for MHC class 1, you're going to have HLA-A, B, C, and each one is going to have so many different subtypes under it. Our class 2 MHC, the way I like to remember it personally, is don't worry, Dr. Dairy Queen has your back. Okay? So DW, DR, and DQ. Is this very high yield? No, but just in case, try your best to remember them. Okay, so as I mentioned to you guys, MHC class 1 are going to be on every nucleated cell in the body except our erythrocytes. MHC class 2 are going to be on our professional antigen presenting cells. What are monocytes? Why didn't I mention them?
Macrophages in the blood.
Exactly. Monocytes are our sleeping macrophages before they actually see something that they have to start phagocytosing. All right, is everything clear in this so far? Are you guys following along? All right, perfect. Now, it is important for you guys to realize that there's a major difference between this type of recognition system and the types of recognition system that antibodies use in order to recognize antigenic peptides. Because antibodies are only going to recognize unprocessed peptides. However, our T cells are going to recognize only processed peptides. They're children, right? They need you to kind of do the work for them. They need you to process those antigens and present them on a beautiful plate and do all of this presentation. So you have to put in more work. Now, looking at the MHC class 1 and 2 structurally, as you guys can see, for MHC class 1, it only passes through the membrane once. MHC class 2 passes through the membrane twice. Also, MHC class 1 is going to have 2 alpha subunits on the top MHC class 2 is going to have an alpha and a beta subunit on top. And why is this important? Why am I mentioning that? Well, it's because this is also the way I like to remember it. When I tell you guys that something has two of the same thing, it means that it is very specific, right? It's going to be like, no, I just want this one. However, when it has two different ones, it can have some sort of stretch to it. It's going to be like, okay, I might accept this more than I accept that. So this type of receptor, which is our MHC class 1, is going to have a lot more specificity. Our MHC class 2 is a bit more lenient. Also, you guys can see that there's this floating subunit here that's not really connected like the others are. That is because our MHC class 1 is going to remain intracellularly as those three subunits, and it's only going to be bound to by a beta-2 microglobulin once it is actually ready to bind to an antigen. So this is not a subunit that gets built in to the receptor. It is one that gets added to it later on. All right. So this portion that I have right here is basically similar to my variable region within my antibody. This is where I actually bind to an antigen. This is where I have my specificity. This is where I have everything that I actually use in order to determine whether or not this is self, this is non-self, this is normal, this is abnormal, what type of antigen is this, what type should I do, all right? So MHC class 1, I mentioned to you guys, was intracellular because it usually will combat or detect intracellular pathogens. I told you guys CD8 positive will be attracted to MHC class 1, and they typically find intracellular pathogens like those of viruses. MHC class 2 also starts intracellularly, but it's going to go extracellularly, and it's going to present on the membrane, and it's going to show that as an antigen presenting cell, it's going to show, okay, here is this extracellular pathogen that I have just found. Do you guys remember this process that I showed you in the previous lecture? What was it called? Phagocytosis, perfect. So phagocytosis plays a very essential role in the beginning of everything that we're going to talk about. So this antigen processing is basically what we can see here, right? So we're going to phagocytose, we're going to do the whole endosome, lysosome, phagolysosome, and then we are either going to release this out, as is the case in a neutrophil, where the free-floating antibodies that we have in the serum can pick them up, or we're going to start presenting them on those, which I didn't tell you guys what are they exactly, but it's either MHC class 1 or class 2. Okay? Now, there's a very helpful video that I think would be really beneficial to show you guys. I hope that the audio works. So just a second, let me share my screen for that. If you don't understand from the video, I'll be explaining everything again anyway, but the way he draws everything out, I think will be very beneficial because he takes you through it step by step. So just let me know if you can hear the audio. Are you guys able to hear the audio? No? Okay, just a second. Are you able to hear it now?
One processing. So what we're going to talk about is how a cell basically processes MHC class 1 together with an antigen.
All right, perfect. Okay.
I'm not doing biology medicine videos.
So since we're all med students, I'm not going to ask if 2 times speed is okay. I hope it is. But I think you guys will thank me for having it on 2 times speed, not anything else. But yeah, there are 2 videos, one for each class. We'll be starting with the MHC class 1 and then moving to the MHC class 2.
Please make sure to subscribe, join the forum and group for the latest videos. Please visit Facebook at Mando Hasurungan. In this video, we're going to talk about major histocompatibility complex class 1 processing, MHC1 processing. So what we're going to talk about is how a cell basically processes MHC class 1 together with an antigen, and how the cell will express this MHC class 1 with an antigen on it. And it will express it to a CD8 T cell, because MHC1 is only expressed when a cell is infected, you can say. So we begin here by drawing the cell. It can be any nucleated cell. Here we have the extracellular fluid, and here we have the intracellular fluid. Some important components within the cell that we have to know is that we have an endoplasmic reticulum, specifically the rough endoplasmic reticulum. And of course, the rough endoplasmic reticulum is close to the nucleus, which I have not drawn. But just know that we have an endoplasmic reticulum. Further, we also have the Golgi apparatus, which is important in MHC1 processing. And lastly, we have an important complex known as the proteasome. Now, the proteasome is within the cytoplasm and has important functions, which we'll talk about soon. So let's begin with the MHC class 1 processing. So what happens is, for an example, is that a pathogen on the outside, it can be a bacteria in this case, will invade the cell, will infiltrate the cell. The pathogen, in this case the bacteria, can, for example, take over the cell. It can multiply within the cell. It can take over the cell machinery if it was a virus. It will essentially infect the cell. And through this process, the pathogen will make its own proteins in the cell. So here I'm drawing endogenous proteins of the bacteria, for example. So these are the protein products of the bacteria. And these protein products, they luckily can enter the proteasome. Now the proteasome is a complex that essentially will degrade these cytosolic proteins into small fragments. So these bacterial endogenous proteins will enter the proteasome, and they will come out in small fragments, about 15 amino acids in length. And so we have many of these. And then these small fragments, bacterial fragments, can then enter the endoplasmic reticulum through what's called the TAP transporter. The TAP transporter is composed of TAP1 and TAP2. And when these 15 amino acid fragments, these antigens, these peptides will enter the endoplasmic reticulum, they can get further broken down to about 8 to 10 amino acids by specific enzymes within the endoplasmic reticulum. So let's stop there and look at the ribosome. Now ribosomes are, as we know, machines that essentially make proteins. And so this ribosome is making proteins, an MHC class 1 protein, which is about 43 kilodaltons in mass. So here it will make the MHC class 1 protein. However, it is incomplete because it is only composed of alpha 1, 2, and 3 domains, as you can see. This incomplete MHC1 is bound to a protein called calnexin. Now, calnexin's function is as a chaperone, bring the MHC1 through the endoplasmic reticulum to prepare it, you can say. Following this, another protein, which is a microglobulin, beta-2M, about 12 kilodaltons in mass, will actually attach to this incomplete MHC class 1, which will essentially complete it. So now we have a complete MHC class 1 made up of alpha 1, 2, 3, and beta-2 domains. Now, at the same time, there will be other proteins attaching on this MHC class 1, specifically calreticulin and ERP57. So what do these proteins do? Well, calreticulin and ERP57 will essentially cover the binding groove, the binding site of the MHC class 1, which will prevent peptides, antigens to interact with the binding site of MHC class 1. Further, the calreticulin and ERP57 will act as a chaperone, and it will bring the MHC class 1 molecule towards the TAP transporter, where the MHC class 1 will interact with another protein called tapicin. When it interacts with tapicin, ERP57 and calreticulin will essentially dissociate, which will allow these small peptide fragments that are in the endoplasmic reticulum, the small bacterial peptide fragments, to bind onto the binding groove of the MHC class 1. However, the MHC class 1 binding site, the cleft, the groove, is very unique and only fits to a specific antigen. And this is because it contains a variable region. The alpha 1 and 2 is unique because it's a variable region and binds to specific antigens. But, for example, if an antigen fits nicely into the binding groove, the MHC class 1, together with the peptide, the antigen, can then leave the rough endoplasmic reticulum. And it will leave the endoplasmic reticulum within an endosome, and it will go through the Golgi apparatus in the normal exocytosis process. And it will then, from the Golgi apparatus, in the endosome, the MHC class 1, together with the peptide, will be presented on the cell surface. So this cell, which is infected by the bacteria, will express MHC class 1 with a small fragment of the bacteria on its cell surface. This is what is happening. And now the MHC class 1 being expressed on the cell surface with the small peptide will then wait for interaction with a CD8 T cell, a cytotoxic T cell. The MHC class 1 will only interact with the cell, and so we can say that it is CD8 T cell restricted. So again, this is our infected cell, and here is a cytotoxic T cell, a CD8 T cell. So the cytotoxic T cell, the CD8 T cell contains a few types of receptors, one of which is this receptor. This is a CD8 receptor, by the way. And it will interact with the MHC class 1 molecule. So what happens is the infected cell will present the antigen, the peptide, to a CD8 T cell. Then the CD8 receptor will check if it's an MHC class 1 being expressed, because the CD8 only interacts with MHC class 1, usually, often. And then the receptor, the T cell receptor's role is to check the antigen, because the T cell receptor only fits to a specific antigen, the peptide. So if both are okay, if the CD8 recognizes the MHC, it's an MHC class 1, and if the TCR fits to the specific antigen presented, the cytotoxic T cell can then release chemicals, cytokines that will essentially kill this infected cell. Before I conclude, I should say that the T cell receptor, the CD8 T cell receptor, contains many CD8 T cells and many T cell receptors, and that this cell presenting MHC class 1 actually will present many types of MHC class 1 on the cell surface, and that each MHC class 1 is unique in that it binds to a specific antigen due to the variable region in the alpha 1 and alpha 2, and that the T cell receptor, each T cell contains a unique T cell receptor. Next, we're going to look at MHC class 2 processing. Thank you for watching. I hope you enjoyed. you have missed videos, please make sure to subscribe, join the following group for the latest videos. Please visit Facebook Armando Hasuna. In this video, we're going to talk about MHC class 2 processing, or major histocompatibility complex class 2 processing. Now, MHC class 2 is expressed only by antigen-presenting cells, such as dendritic cells, B cells, and macrophages. Now, the MHC class 2 will have an antigen on it and will present it to a naive CD4 T cell. Let's look at the process in an overview. So this here is a cell. Here is the extracellular fluid, and here's the intracellular fluid of the cell. This cell can only be an antigen-presenting cell, so let's just say it's a macrophage. Now, within the extracellular fluid, there can be pathogens, such as a bacteria here. The macrophage, knowing that this is a pathogen, will engulf the pathogen, the bacteria, through a process called phagocytosis. Let's stop there for a moment and see what we can find in the cell. So within the cell, this is not a mitochondria, it's actually an endoplasmic reticulum, rough endoplasmic reticulum, where we can find ribosomes and where protein packing occurs. So here we have the endoplasmic reticulum, here we have the Golgi apparatus, and most importantly for MHC class 2 processing, we have lysosomes. Now the lysosomes contains acids, and so within it is an acidic environment. Now back to the bacteria that was phagocytized. The antigen-presenting cell, the macrophage, will destroy this bacteria by allowing the fusion of both the lysosome and the phagosome, like so. And this will form what's called the phagolysosome, which has an acidic environment, about pH of 4, and this is due to the lysosome. And because of the acidic environment, this will cause the content, being the bacteria, to break down, leaving various bacterial peptides or antigens here. Now within the endoplasmic reticulum, the ribosomes are synthesizing MHC class 2 molecules. The MHC class 2 is made up of alpha 1 and 2 domains and beta 1 and 2. The alpha portion is about 35 kilodaltons in mass, and the beta is about 23 kilodaltons. The MHC class 2 will have another protein interacting with it as soon as it's synthesized. This protein is called II protein, better called actually, I think it's the CD74, which will prevent endoplasmic reticulum proteins from binding onto the MHC class 2 groove. So it will prevent these sort of peptides from binding onto the groove here. The II structure, protein, also acts as a chaperone, bringing the MHC class 2 out from the endoplasmic reticulum in endosome, then through the Golgi apparatus, and forming a new endosome. This new endosome is slightly acidic. The acidified endosome actually degrades the II protein, leaving a fragment called clip on the binding site, or the groove of the MHC class 2, thus allowing no peptides or antigens to bind onto MHC class 2. Following this, another protein called HLA-DM will interact with MHC class 2, releasing clip from the binding groove. The phagolysosomes containing bacterial peptides will then fuse with the MHC class 2 endosome, and this will allow the peptides to bind onto the groove of the MHC class 2. However, only a specific peptide, specific antigen is able to bind onto the groove of the MHC class 2. And this is for any MHC class 2 because it's all unique. So after a specific antigen will bind onto the groove of the MHC class 2 like so, the macrophage, which is an antigen presenting cell, will express it on its cell surface. So here, this is MHC class 2 with an antigen. The MHC class 2 is expressed and will wait interaction with a naive CD4 T cell. The naive CD4 T cell is MHC class 2 restricted, which means that it requires interaction with an MHC class 2 molecule to become activated, basically. When the naive CD4 T cell becomes activated, it will become a T helper cell, and this is very important in promoting the immune response. So let's see what sort of interaction occurs between the antigen-presenting cell and the naive CD4 T cell. So here I'm drawing out the cell membrane of a naive CD4 T cell, which essentially is a T helper, naive T helper cell. What first happens is a receptor called CD4 receptor, hence it's a CD4 T cell, will check the MHC molecule. It will check if the MHC being expressed is an MHC class 2, because CD4 receptor only interacts or recognizes MHC class 2. Now, the CD4 receptor is made up of segments D1, D2, D3, and D4. It will actually bind onto the alpha portion of the MHC class 2. If that's okay, the T cell receptor, TCR, will then check the antigen. Now the T cell receptor's binding groove also, it only fits to specific antigens as well. So let's just say in this case, the TCR can fit to this antigen being presented by the MHC class 2. So if the CD4 receptor will check the MHC class 2 and confirms it, and the TCR checks the antigen and confirms it, this will trigger a cascade of events within the naive CD4 T cell. The signal transduction, the cascade, will eventually lead to the activation of the naive CD4 T cell to become a T helper cell. And so this T helper cell can then promote the immune response by activating B cells, for example, or other things. Of course, it's not that simple, this activation process. In order to activate the CD4 T cell, naive CD4 T cell, other receptors such as B7 has to bind to another receptor, as well as there's release of cytokines. But that's too much detail. I hope you understood the basics of this. Thank you for watching.
That video for you. Did you find that video beneficial, you guys? As I said, I will be going over the entire thing again myself, but I just thought it would be good for you guys to see the step by step, the way it was drawn, so that you can follow along with everything. I know it had a bit more than you guys need to know, but I just thought it would be helpful so that you can get a full picture of everything. All right. So I want you guys, now that you have seen this video, to help me out so that we can get through those even faster. As I mentioned to you guys, we need to have some antigenic processing for everything that we want to present to our T lymphocytes. whether that is on MHC class 1 or MHC class 2. So our antigens are going to go through multiple phases, let's say, until they're able to get presented on this peptide. So when I'm talking about MHC class 1, I'm typically talking about, as I told you guys, an intracellular sort of pathogen. Now, this can be from within the cell itself. It can be some sort of tumor. It can be some sort of, like, let's say, a mutation, or it can be, let's say, a virus that injects itself into the host DNA and then it begins to replicate consistently. Now, this type of infection is going to go to a proteasome, which is a cylinder that is made up of multiple proteolytic enzymes which degrade ubiquitinated proteins. What ubiquitin is basically a tag onto those proteins that says those are not mine, those are not normal, they should be degraded, they should be taken away from me. So a tagged protein with ubiquitin is going to go through the proteasome, it's going to get broken down. Once it gets broken down, simultaneously, we also have this protein synthesis of this MHC class 1. It's consistently getting built up, and if it's not presented with any antigen, it will get broken down as well. However, since I do have an antigen here, it's going to bind and it's going to get presented. Now, what is the process in order for it to bind and to get presented? First of all, we are going to be having our MHC class 1. It won't be coupled with beta-2 microglobulin just yet because it is not yet presented and it's not yet ready, right? However, as it is getting through this endoplasmic reticulum, as it is moving through it, the chaperone proteins will basically take it along the way and put a beta-2 microglobulin along with it. Those chaperone proteins are also super important because they act in order to hide the antigenic portion of this MHC, or where this antigen is supposed to bind. Those types of chaperones, the very important ones for you guys to know, are the ERP57 and the tapicin. Now, tapicin, the name of it signifies something that's important. So tap, I want you guys to imagine like the antigen is almost knocking on the door of the endoplasmic reticulum. It's tapping on the endoplasmic reticulum to enter. So tapicin is super close to the TAP1 and TAP2, where our antigen will actually enter in order to bind to our MHC. So once tapicin joins with our MHC, and once we finally are able to get this antigen to present, this entire MHC is going to go through the Golgi apparatus, get packaged into an endosome. This endosome is going to fuse. The membrane of it, at least, is going to fuse with the lipid membrane of the cell, and it's going to get presented as a peptide-loaded MHC class 1. So the antigen is actually going to travel all the way from the endoplasmic reticulum through the vesicle and to the cell surface along with our MHC. Once this MHC class 1 is presented, our CD8 positive cytotoxic T cell will recognize it and realize something is wrong here. I should not be seeing this type of peptide. I should not be seeing this type of antigen. It should be normal, right? So it is going to perform its cytotoxic ability and kill this cell. Is that clear for everyone? Is MHC class 1 clear?
Doesn't cytotoxic T cells kill cells that don't have MHC? Or is that?
No, the natural killer cells are going to recognize cells that have an absent MHC and kill them. Because you're always supposed to be presenting something on MHC, right? This is on all nucleated cells. So you usually just present protein, like degraded proteins that are normal from within the cell itself. So a cytotoxic T cell is going to be consistently surveillancing, and this is what we call actually immunosurveillancing. They're constantly moving around in the blood, and then once they recognize an abnormal MHC, they're going to realize, okay, something is wrong with the cell, it needs to be dead. If there isn't an MHC class whatsoever, then cytotoxic T cells will not recognize that. They're going to be like, okay, I don't see anything abnormal here. However, natural killer cells will realize, no, it should be presenting me with something. As I told you guys, it's like that bouncer that basically checks for an ID. So it's like, no, you don't have your ID. Even if your ID is fake, you don't have it whatsoever. So it goes and it kills it immediately. Is that clear? Does that make sense?
Yes, thank you.
All right, for sure. What is ERP57? It's a protein, yes, it's a chaperone, which basically, as I told you guys, is going to continue to hide the alpha 1, alpha 2 portion in order to prevent the MHC from binding to intracellular antigens that are normal. And then if they present them, you're going to get this cascade of an autoimmune reaction where you're going to be attacking a cell that doesn't actually need to get attacked. So those chaperone proteins are very important. And first of all, hiding that site. Second of all, moving this MHC towards the tap and towards where we actually have this antigen so that it can bind to it, as well as providing this beta-2 microglobulin so that it can bind and give me a complete MHC. Any other questions? As you guys have probably seen in the previous presentation, I'm including everything in the notes. So don't bother to take notes or like worry. Everything that I say is in the notes, and everything that you need to know, inshallah, is either in the slides or in the notes. So I told you guys that this is MHC class 1, and I'm going to present all of this. What type of cell does this happen in particularly? Because I mentioned it's a viral type of thing, right? So can you guys imagine or think of which type of cell would do all of this presentation? All cells except white blood cells. That is true. All cells do present MHC class 1, but which ones would particularly present me with an abnormal MHC? I haven't mentioned it, so it's completely okay if you don't know. I'm just trying to see if you can figure it out. Antigen presenting cells, yes, perfect. Which of them did I tell you guys is the most paranoid? CD56, perfect, yes. So our dendritic cell is going to be super paranoid. It's going to be like, no, even if this is getting divided intracellularly, even if this is a protein I'm producing, I don't think this is right. This doesn't seem right to me, so it has to be wrong. So our dendritic cell is going to present this abnormal antigen on MHC class 1. And not all antigens are presented to dendritic cells, though. Not every single one of them is going to come to a dendritic cell. So how does it happen that dendritic cells are able to always show this abnormal MHC? The way it does that is basically through this cross matching. It takes over from other cells. They know to go to their grandpa. They know, like, he knows best. I'm going to go to him. I'm going to ask him, is this abnormal? Is this weird? I don't know. So other cells are actually going to go to dendritic cells and give them the abnormal antigen that they have, and then the dendritic cell is going to present it on MHC class 1. So just remember it as this grandpa that everyone goes to for advice, and because of its dendrites or its very long legs, it can reach multiple cells and it can extend very, very far. Is that clear for everyone?
So dendritic cells can have both MHC 1 and 2?
Yes, exactly. Dendritic cells can present both MHC class 1 and MHC class 2, almost simultaneously even, because they can take those antigens from other cells, which were intracellular to them, but are now extracellular to the dendritic cell, and they can also take extracellular pathogens, and they can present both. So dendritic cells present both MHC class 1 and MHC class 2. In fact, if you want to really think about it, all our antigen-presenting cells should have both, right? Because MHC class 1 is going to be present on all nucleated cells, and MHC class 2 is going to be on all antigen-presenting cells. So our dendritic cells, our B lymphocytes, and our macrophages will express both of them. All of them are going to express both. Does that make sense? All right, perfect. Now let's move on to MHC class 2. MHC class 2 is when I have an extracellular pathogen. So think of it as a bacteria. Think of the actual phagocytosis figure that I showed you guys earlier. Now, we had a proteasome in MHC class 1. However, in MHC class 2, we're going to have a phagosome. So this phagosome, after phagocytosis, will fuse with a lysosome to give me a phagolysosome. And then it's going to process the antigens. Try to remember the picture with me. It processes the antigens. It either sends them out, as a neutrophil does, or it presents them on MHC class 2. Now, the way that it's going to present them on MHC class 2 is, once again, within the endoplasmic reticulum, we are still producing this protein because it's a protein like any other. However, our MHC class 2 has all 4 subunits already. It doesn't need an extra subunit to come to it. It doesn't need anything to help it. But we do still need to hide that region where it binds to the antigen. So for MHC class 2, similar to how we had all those chaperones, we are going to have what is known as II. II is going to bind to MHC class 2 and hide its antigenic binding portion. As this goes through the Golgi apparatus, we still have not met an antigen, right? For MHC class 1, we meet the antigen within the endoplasmic reticulum. Here, we still have not met one. It goes through the Golgi apparatus, gets packaged into an exosome, and then within that exosome, that II that I told you guys about is going to get clipped, and only some of it exactly at the antigenic binding portion is going to remain as a clipped protein. The rest of it is going to be dissolved. Now we have this phagolysosome. We have this antigen that's ready. This phagolysosome is actually going to fuse with our exosome where we have this MHC class 2. Once that happens, we need to be able to have an available antigen binding site. So HLA is going to come, not the same ones that we have been talking about this entire time, not the, what did I tell you guys? Don't worry, Dr. Dairy Queen has got your back. We're gonna have HLA-DM. Okay, so HLA-DM is going to remove this clip protein and allow the antigen to bind to our MHC class 2. This MHC class 2 is going to go to the cell membrane and be presented where a naive CD4 T helper cell will find it, recognize it, mature, and be able to carry on with its function. Is that clear for everyone? All right, all clear, perfect. Now, looking at the actual response from the CD4 positive T cells and CD8 positive T cells, as in the video, in order for you to actually be able to bind, you have the CD8 receptor, which will bind to CD8 MC, what is happening? MHC class 1, and then CD4, which is going to bind to MHC class 2. All right, so 8 times 1 is 8, 4 times 2 is 8. We always have to have an 8 somewhere. To quickly review everything, our CD4 positive and our CD8 positive T cells, they need everything to be presented to them on a plate of gold. So very briefly going over everything again, an MHC class 1 type of receptor will meet the antigen within the endoplasmic reticulum itself, and then it's going to get presented. And MHC class 2 is going to meet the antigen after it is already in an exosome that is about to be sent out. It is going to fuse with the phagolysosome where we have this antigen and it is going to be presented. Our MHC class 1 only passes through the membrane once. This is wrong, it should be twice. Our MHC class 2 passes through the membrane twice, and it has 2 alpha and 2 beta subunits here. This has an alpha and a beta, so this is more specific. Is that clear for everyone? All right. Now, I told you guys what's the marker for dendritic cell. How old is our grandpa? He is 56 years old. So all of their ages are their markers. And all of the markers that I mentioned are the ones that you need to know. What about natural killer cells? Natural killer cells are CD56. To my knowledge, it's dendritic cells that are CD56. Natural killer cells, as far as I'm aware, are 30 something, but I will check and I'll get back to you guys about that for sure. But even in my preparation, I remember reading it as CD56 for dendritic cells. But if you guys have it in your slides as that, then I might have misread it. So I'll check and get back to you guys about that point for sure. All right. Any other questions? All right, perfect. Okay. So I told you guys that a virus is going to infect a cell and then it is going to start to replicate within the cell and it's going to basically hijack the machinery of that cell, right? And in the previous lecture, I was telling you how sometimes the virus, because it's being sneaky, is like, oh, well, you don't have to present an MHC. Nothing's wrong here. I'm not an antigen. And then the natural killer cell is going to recognize that and be like, no, wait, hold up. I should be seeing MHC class 1, even if it's just of a degraded protein. So the virus is going to try to evade the immune system through the removal of a newly synthesized MHC molecule from the endoplasmic reticulum by preventing the successful folding of a newly made MHC protein, by inhibiting the transcription of MHC genes, by blocking the peptide transport by tap. So I told you guys it's like they're knocking on the endoplasmic reticulum or they're tapping on it. So if you don't have this tap or you're preventing this tap, the antigens are not even going to go into the endoplasmic reticulum where they typically meet this MHC class 1. However, as I told you guys, we have a backup system, which is our non-self hypothesis. Who is going to go against this? Who's going to go against a lacking MHC?
NK cells.
Exactly. So our natural killer cells are going to kill any cell that does not have an MHC class 1. And that brings us to the end of the last lecture that you guys have. Do you have any questions for me? We have practice questions, which is the last section, but do you have any questions for me at all before we get to those? All right. Perfect. Okay. So now for the practice questions portion, I have included a question and its explanation right after, and I'll keep that as well for the slides I actually sent you guys. And I want you all, every single one of you, to send me a message, whether that's to everyone or to me privately, of what you think the answer is, just so that I can make sure that the topic is fully covered, no one has any confusion in anything whatsoever. So let's get started. I want this to be you guys, okay? You should know all the answers. Starting with question 1, credits to your exam committee and then my personal batch's exam committee. But here we go. Question 1. Would you like me to read the question or do you just want to read it yourself and tell me the answer? All right, I'm getting our A's, which is hopefully what you guys are going to get in this midterm as well. A. Okay. Does anyone think it's anything that is not A? Does anyone have any doubt about what else it could be? All right, I think we're good. So we need to have this specific type of cell that responds specifically to microbes and also exhibits memory, which is none other than our B cell. You guys are completely correct. Our naive B cell can become a memory B cell, or it can become a plasma cell that actually secretes those antigens against the microbe, or it's a long-lived memory cell that gets stored, and then your body can know that you've had this infection before. Next one. All right, so far I'm only getting E's. Does anyone else have a different answer? It's completely okay if you give a wrong answer. It's gonna actually help me make sure that I cover the thing that you are doubting. So please don't be afraid to make a mistake. C, okay. A, okay. Yeah, see, I thought this question would be confusing. If I was in your place, I would definitely be confused, which is exactly why I got it here. Because macrophages are, I got an F somewhere in the chat, which is absolutely amazing. That would be my answer. So good job. That's how Al-Faisal functions. So macrophages are an interesting bunch because they have so many different mechanisms upon which they work, right? So they can either be tagged for opsonization using an Fc receptor, they can react to an actual membrane immunoglobulin through their Fc receptor, they can have pattern recognition receptors, they can have complement receptors, they can have antigen receptors, they can have all of those. So it's very confusing. But the question says they recognize the microorganisms themselves. So they're not recognizing another type of immunoglobulin, so that removes C and D. And they're not recognizing another type of protein, so that removes A and B. And that just leaves us with E, which is a pattern recognition receptor. Going back to our slide, when you want to recognize non-self, you typically, and that is the most common way that they use it, you use a pattern recognition receptor or a PRR, and the types that we have are our node-like receptors, our toll-like receptors, and our rig-like receptors. Is that clear for everyone? Does anyone have any doubt about it? All right, perfect. So when I spoke to Dr. Garwin, he actually said that I should just get you guys a bunch of SAQs, but I tried my best to get you MCQs of varying levels, some of which are super simple, some of which are a bit more complicated, but all of them are the type of questions that Dr. Garwin would get. So it's good to try to familiarize yourself with them. Next up. What was TLR aureus LPS? A, okay. I don't know. I think this is foreshadowing, honestly. Whenever the answer is A, you guys are all immediately there. You just get it immediately. So good job. Yes, the answer here is A. And looking back at our slide, it is because when a molecule is too small, it is an antigenic molecule, but it is too small to kickstart a response. It is called a hapten, and it needs to bind to a carrier protein in order to stimulate this clustering and this actual activation. All right, next. All right, everyone is saying C. Does anyone else have a different answer? Okay. If I were to change C from being variable region to being FAB region, would that still be the answer? Perfect, you guys. Absolutely wonderful job, mashallah. All right, so our FAB region is also our variable region within our antibodies, and it's where we have our CDRs, it's where we actually have the binding of the antigen, and this is where we have all of this hypervariability. Next step. All right, see, I told you guys, whenever it's in A, you get it immediately. MashaAllah, great job. So going back to our slide, the affinity is the strength of the binding, the avidity is the number of interactions and the capability of binding. Now, what is valency before I move on? How can I define valency? The number of binding sites. Okay, so it's basically, you are correct. It's the number of occupied binding sites, to be very specific, because we can have two binding sites, right? But only one of them is actually occupied. So this would be a monovalent. However, when they're all occupied, this is bivalent, and we can see this in IgG, and what other two? Where can we see this bivalent sort of interaction? IgE and IgD, perfect. This polyvalent is very specific to IgM. Next question. I might actually just show you both at the same time. So you just like type out the actual answer so I know which one you're referring to, or just put it as like 6, 7. I think you okay. All right. Perfect. One of you wrote G for IgG in the list. I get it. Okay. 7C. Okay. So 6 is A and 7 is C. Yes, perfect. Good job. So our immunoglobulins are divided into 5 types. IgM, I told you, is the mother. It's the primary response, but it's not very specific. IgG is the grandmother. They have better aim, so they're in the secondary memory response. They're a lot more specific, and they can cross the placenta in pregnancy. IgA for abundant, and it's always shed in the bodily fluids. IgE, egg, eosinophil, allergic, and IgD is mostly membrane bound, and you don't really have to remember it. Next step. There's no way you read that answer that quickly. Read everything. Just read them. It's okay. See, when there's a question mark after your answer, I just know, like, you got the right answer. Good job. But I don't want you to have that sort of hesitation during the exam, so I'll be going over this again. In the context of the adaptive immune system, which of the following accurately represents the role of RAG1, RAG2, and TDT enzyme in VDJ recombination? Let's go to the slide and then get back to this. So our VDJ is this haphazard rearrangement of our V, D, and J regions, which is done using RAG1 and RAG2. They are the DJs, right? Because they start with D and J and then they add the V. They remix this DNA gene and they're going to give me the actual product that I want at the end. And then TDT is going to just come in and have some random deletions, some random additions to add even more variability. And this is where we have most of our variability. This gets transcribed into precursor RNA, and after splicing, it's mRNA, which gets continuously divided. We said that RAG1 and RAG2 have the combinatorial diversity, which is basically because we combine different VDJs, and we also combine different either kappa or lambda for the light chain. And we said that TDT has the junctional diversity because at the junction of those VDJ regions and the actual constant region, which determines if it's IgM, they're going to have a function and they're going to increase the variability. Now, going back to our question, looking at C, it says RAG1 and RAG2 enzymes mediate VDJ recombination to generate antigen receptor diversity. Is that correct so far? Yes. While TDT enzyme adds non-template nucleotides to enhance receptor viability. Does that make sense? All right. Very briefly, let's go over why the rest are wrong. RAG1 and RAG2 catalyze the joining of heavy and light chain genes. Do they do that? Do they join the heavy and the light chain? All right, so this is automatically incorrect. TDT enzyme also does not mediate class switching. That happens through T-B cell interactions. B says that RAG1 and RAG2 enzymes catalyze the recombination of T-cell receptor genes. Does it do that for T-cell receptor genes? Or are we talking about antibodies on B cells this entire time? All right. And it says that the TDT enzyme facilitates somatic hypermutation of antibody genes. Is that correct? That's a big no. Why is it a big no? What is the function of TDT enzyme? Okay, so before saying it's a big no or a big yes, think of the actual function of the enzyme. And you were correct initially, so don't doubt yourself. It does not facilitate the mutations. It just adds the like the nucleotides. It adds and takes away the nucleotides. It doesn't contribute, let's say, to the mutation by just stimulating something to happen. No, it does the action itself. C, we said is correct. D, RAG1 and RAG2 enzyme facilitate antibody production in plasma cells, while TDT enzyme is responsible for somatic recombination in memory cells. Don't even get me started on the amount of things that are wrong here. Plasma cells are the activated B cells, so we need to already have a B cell to have a plasma cell. And in memory B cells, you're not going to have somatic recombination because that would defy the entire purpose of a memory B cell. You need to have the actual exact antibody so that if you have a secondary infection, you can present, you can show the body that, okay, I know what this antigen is. Finally, for E, RAG1 and RAG2 enzymes play a role in allelic exclusion, while TDT enzyme ensures receptor specificity for a particular antigen. Now, allelic exclusion was something that I did mention to you guys. Do you remember what that was for, though? Was it for RAG1 and RAG2? Okay, no, what was it for? It's for the VDJ regions. Okay. Exactly. Perfect. It only comes from one chromosome. So we get the gene from both of our parents. One of them gets activated and the other one remains active. Gets inactivated, sorry, and the other one remains active, and this is called allelic exclusion. Great job, you guys, mashallah. Let's move to our next question. All right, perfect. What other opsonin do we have from our complement cascade? C4b, exactly. Next question. All right, great job, everyone. Yes, that is correct as well. Because acting as a chemokine basically also means everyone is saying C, but which is C though? I showed them in order. So this is C and this is D. So let's apply that to the actual slide. I was asking you guys about opsonization and I was asking you guys about chemotoxins. So chemotoxins are our anaphylotoxins, which are C3A, C4A, and C5A. Remember, A for anaphylotoxin. Our opsonizing proteins or our opsonins, our C3b and our C4b, which are binding to this antigen and presenting it to the macrophage. Is that clear for everyone? All right, perfect. Next question. We're almost done, you guys. I just wanted to include as many questions as possible so that you feel confident. And I know you don't have an actual session on immuno questions, so okay, two people are saying B. Does everyone agree or does someone have a different opinion? All right, so you're all saying C. Oh, no, sorry. Some of you are saying B and some of you are saying C. Okay, let's go over it before I tell you which one's the answer. If we're saying C, we're talking about direct lysis of bacterial cells. What makes this happen? What directly lyses a bacterial cell? Okay, natural killer cells. But did I tell you guys that natural killer cells act on the antigen? Or did I tell you guys that they act on the normal body cells, let's say, the somatic cells, the body cells that have an absent MHC or they're cancerous, they're tumor cells. So they are actually from within the body. They're not pathogens. Okay. So what causes this direct lysis of bacterial cells? What was something that would bite the bacterial cell and cause it to flood with water and swell up and burst? Exactly, our membrane attack complex. MACPAN, yes. So, what triggers this MACPAN? Because that's the final product, right? The complement pathway, exactly. So through complement activation, am I only allowing myself to have this membrane attack complex, or am I providing the body with multiple different ways to combat this bacteria? I can also do what? I can also do opsonization. I can also lead to a hypersensitivity response, right? So through complement activation, this is a lot more protective than if I were to only use the membrane attack complex. Does that make sense? Now, opsonization is once again same thing because those two are the end products of complement activation. Antibody-dependent cell cytotoxicity. Yo, this is specifically for natural killer cells and is once again just one possible outcome. Then neutralization of bacterial toxins is used, as I told you guys, for vaccinations. It's used when a virus is completely coated with the antigen, the antibody, sorry. So in those cases and everything else, I only have one protective outcome, right? But if I'm activating the complement cascade as a whole, then I can activate so many different protective outcomes that can ultimately kill so many more bacterial cells. Does that make sense for everyone? All right, so looking at our slide, we had our pathogen. Then immediately we would bind this antibody, which is IgM or IgG to it. Then this would stimulate the entire classical pathway. And then eventually we would get our membrane attack complex. So it is an end product. Okay? Oh, dang, dang. What's happening? Oh, no. Okay. Next question. All right. So I got DD and then just someone being upset. So what do you think it is? Or why are you confused? I mean, I understand looking at microbiological terms might be a bit off-putting, but MHC class 1, take it this way, take it backwards. MHC class 1, if I'm presenting a microbe on it, I'm not presenting something that is from the body itself, then it has to be something that is intracellular. And what did I keep on telling you guys is intracellular? Viruses, right? This is incorrect. It shouldn't be repeated twice. But yes, viruses. So either one of those would be the correct answer because it's the same. Oh no, Haemophilus influenza is a bacteria. Haha, sorry. Influenza virus. Yes. So looking back at our slides, we're going to have intracellular pathogens like viruses present on MHC class 1. Okay? Last question, you guys. Three, A, okay. Anyone else have a different answer? Someone's going all in on E. Someone is saying B. Okay, so the person going all in on E, I think they're just trolling, but good on you. So for CD152, even I don't know what this is for, honestly. Someone is saying B. I understand why you'd say that. But if I am talking about an intracellular pathogen, once again, I'm referring to CD8, right? But since CD8 is not one of the options, I'm not just going to go appeal the question immediately. Let me try to think of what other marker does CD8 have? What other marker do all T lymphocytes have? And that is CD3. CD3 presents first, and then afterwards, you're going to have a CD4 and a CD8 cell. So looking back at our slides, we see that we have a common CD3 positive progenitor that then can give me CD4 positive or CD8 positive T cells. And with that, we finally come to a close. Does anyone have any questions about any of the lectures? Anything at all? If something isn't clear, if there's something you'd like me to explain again, if you feel like you might not 100% nail something, now is the time. I don't mind going over everything again. Literally, if I have to do the entire thing again, I don't mind. Everyone can leave and I could teach you the entire thing again. Any questions whatsoever about any of the lectures? Innate immunity, complement, MHC, immunogenetics, because you guys, the most requested ones were immunogenetics and MHC. So I really, really, really hope that I was able to deliver those in a very concise way, in a very clear way, and everything is clear. Any questions at all? All right, before you all start leaving, please scan this QR code to give us your feedback. Hope we'll see you in future crash courses like this. I mean, if you guys liked my explanation, it was actually beneficial for you, then yes, inshallah. Just let me know whenever you guys need any help. So I guess no more questions. Yes, you can stop the recording.